Good morning, students. Today we'll discuss about WBC. WBC, they are known as white blood cells because of their color, and they are the real fighters and soldiers of the body. The WBCs or the leukocytes, they are the cells of immune system which are involved in defending our body against both infectious diseases as well as the foreign material. 5 different types of leukocytes they are present. They are formed from the bone marrow by a process known as leukopoiesis. They are formed from hemopoietic stem cells. The WBC, they live for about 3 to 4 days in average human body. And WBCs, they are found throughout the body. They are present in the blood as well as the lymphatic system. In this diagram, you can see the blue colored cells, bluish colored, they are the WBCs, whereas the pinkish colored cells, they are the RBCs. Now coming to the functions of eosinophils. Following activation, eosinophil effector function includes the production of cation granule proteins and their release by degranulation. Then reactive oxygen species such as superoxide, peroxide and hypobromide. Enzymes like elastase. Then cytokines like interleukin 1, interleukin 2, 4, 5, 6, 8, 13 and tuber necrotic factor alpha, they are released. In addition, eosinophils play a role in fighting viral infections, which is evident from abundance of RNAs as they within their granules and in fibrin removal from during the inflammation. Eosinophils also with basophils and mast cells are important mediator of allergic responses and asthma pathogenesis and are associated with disease severity. They also find helminthes colonization and can be slightly elevated in the presence of certain parasites. Now coming to the basophils. Basophils appear in many specific kinds of inflammatory reactions, particularly those of the allergic symptoms. Basophils contain anticoagulant heparin, which prevents blood from clotting too quickly. Then they contain vasodilator, that is histamine, which promotes blood flow to the tissues. They can be found in unusually high numbers at the site of ectoparasite infections like ticks. Like eosinophils, basophils play a role in both parasitic infections and allergies. They are found in tissues where allergic reactions are occurring and probably contribute to severity of these reactions. Basophils have protein receptors on their cell surface that bind IgE, that is immunoglobulin E, and immunoglobulin that is involved in macroparasite defense and allergy. It is the bound IgE antibody that confers a selective response to these cells to environmental substances, for example, pollen proteins or the helminthes antigen. Now see what is the role of the neutrophils. The foreign invader signals nearby the neutrophil to squeeze through the endothelial cells that lines the blood vessel and enter the infectious tissue, right? So this process, the neutrophils, they change into macrophages and they perform the process of phagocytosis. They ingest the bacteria and release toxic products that kills the bacteria. Chemotaxis. Neutrophil undergoes a process known as chemotaxis, which allows them to migrate towards the site of infection or inflammation. Cell receptors allow neutrophils to detect chemical gradients of molecules such as interleukin 8, interferon gamma, the IFN gamma, which these cells use to direct the path of their migration. Neutrophils have a variety of specific receptors, including complement receptors, cytokine receptors for interleukins and interferon gamma, receptor for chemokines, receptor to detect and adhere to the endothelium, receptors for leptins and proteins, and Fc receptors for the opsins. So see here in this figure, you can say that the neutrophils first of all bind with the protein present on the endothelium. Then L-selectin is attached to it. And then what happens? The shedding of L-selectin is there, and the neutrophil, it is bound with the help of integrin to E-selectin. Adhesion is there with the help of integrin, and ultimately there is a process known as diapedesis. Diapedesis is a very important process. It is known as the coming out of neutrophils from the endothelium through the narrow spaces into the infected sites. So here what happens, the complement tissue, that is C3a and C5a, they are very important along with chemokines and histamines. Prostaglandin 2 plays very important roles along with the leukotrienes. Then these neutrophils, they'll come to the infected areas, and with the help of phagocytosis, the destruction of C3b coated bacteria is there. So this is antimicrobial function of the neutrophils. Neutrophils express and release cytokines, which in turn amplify inflammatory response by several other cell types also. Neutrophils have three strategies to directly attack the microorganism. Number one, phagocytosis, as we have seen, that is directly ingesting the bacteria. Second, release of soluble antimicrobials, that is the granuloproteins. Then third is the generation of neutrophil extracellular traps, that is nets. So you see here, in this way the neutrophils, they are helpful in fighting with the infection through the fight. These are the cells where the neutrophils, they are present, and they'll ingest the bacteria. So phagocytosis, neutrophils, they are phagocytes capable of ingesting microorganism. They can internalize and kill many microbes. Each phagocytic event resulting in the formation of phagosome, into which reactive oxygen species and hydrolytic enzymes, they are secreted. The consumption of oxygen during the generation of reactive oxygen species has been termed as respiratory burst, although unrelated to respiration or the energy production. The respiratory burst involves the activation of enzymes, that is NADPH oxidase, which produces large quantity of superoxide or reactive oxygen species. Superoxide dismutase, spontaneous or through catalysis via enzyme, are known as superoxide dismutase to hydrogen peroxide, which is then converted into hypochlorous acid, that is HCIO, by green heme enzyme, that is myeloperoxidase. It is thought that bactericidal property of HCIO are enough to kill the bacteria phagocytosed by the neutrophils, but they may instead a step necessary for the activation of proteases. Now see here in this is a neutrophils trapping the bacteria. So microbes adhere to the phagocyte. Phagocyte forms pseudopodia, which eventually engulf the particle. Phagocytic vesicle containing antigen is known as phagosome. This phagosome is fused with lysosome to form phagolysosome. Here the enzymes, they act on it and they kill the bacteria, digest the bacteria, and the residual body is formed. The useful things, they are absorbed, and later the residual body is thrown out with the process known as exocytosis. So my sincere thanks to www.google.com for providing us beautiful pics so that we can explain our topic well.